Good morning, Hyderabad. I wish to thank Express Pharma, Viveka, for inviting me to share some thoughts with this conclave. Formulating a program for formulation development of complex products. That's the topic I wish to share some thoughts on. In collecting the information I thought would be useful to share with you, I reflected back on my experience leading the Sandoz biosimilar development program along with taking on complex generics for Sandoz Biopharmaceuticals, including generic enoxaparin, generic Copaxone, leading the efforts on next generation product assessment at Philip Morris International, a situation which was more highly complex and uncertain, particularly with respect to the regulatory requirements for modified risk tobacco products for approval in the US by US FDA. A year at Vocard, and then my practice, consulting and advisory practice in India since July of 2013. The key point that I wish to share with this audience of senior leaders of formulation development in India is to think about complexity in a way that allows you to reduce uncertainty and allows you to leverage complexity. Complexity is something that is a way of doing business going forward, and the quickly you build your organization to deal with complexity, the more successful you will be. So, with that in mind, just thought it'll be important to emphasize that there are three themes that need to be considered in an integrated manner. Taking formulation development from art to science. What does that mean? It means providing scientific evidence and rationale for why a particular formulation is being proposed and developed, and why and how and what is the basis for claiming sameness or equivalence or similarity. And by science means it has to be a test of hypothesis. A test of hypothesis can be in vitro. A test of hypothesis can be stability study. A test of hypothesis can be biostudy or a clinical trial. Given the complexity of products and the evolving regulatory requirements, complexity of the process, you have to think of this as a program to plan ahead to address how during development you will deal with emerging and evolving regulatory requirements for the product you're going to develop. And so you need a level of agility, you need a level of openness, and not a fixed mindset of time to exhibit batches, time to bio batch, and so forth that a typical generic organization works under. There is a science of complexity, and you can utilize it to leverage and reduce uncertainty and to leverage complexity. And I'll share some elements of that during the course of this discussion. Let's remember, we use terms that we often only think everybody else knows the same meaning as we do. Therefore, I'm just taking a moment here to share with you the share with you the dictionary definition of formulation, program, and complex. And as you look at that, you'd relate to some parts of it or relate to all of it. But it's important to understand that we speak in terms that we don't assume people will have the same level of understanding that we do when we talk about something. Formulation, a material or mixture prepared according to a particular formula. Program, a planned series of future events, items, or performance. Complex consisting of many different and connected parts. That's not just what makes things complex. It's the non-linearity, it's the interaction, it is the emergent properties and the emergent requirements, and collectively makes the system unpredictable. So you have to reduce complexity as part of your development program. Today, many people talk about complexity crisis. In um, the consumer product world, you can see innovation, continual improvement in product offerings, functionality. You see rapid introduction of new technologies, and by virtue of uh, uh, the sheer volume of functionalities, sheer number of functionality, number of changes, number of innovation, there is a level of complexity to get the organization be aligned with the new quality target product profile, such as your cell phone and so forth. In the pharmaceutical world, I think we have to think about analytical methods, the regulatory requirements for evidence, and and everything associated with their predictability, I think, is an important element. Complex product systems contain many components and layers of subsystems with multiple nonlinear interconnections that are difficult to recognize, manage, and predict. Here, my emphasis will be on the modern analytical technology portfolio you will bring to bear on your characterization of your reference product to establish sameness and to provide the evidence in the most efficient way of sameness or biosimilarity without having to rely on costly, expensive, and often blunt clinical trial data. That is the key thing. And if you remember the way we define formulation, a particular composition, over the years we have talked about that being qualitatively and quantitatively the same, Q1 and Q2. The complexity we have today is Q3, which is the matrix and how that matrix relates to performance, characterization of the matrix, which are interactions among particles and interactions that can be ionic, that can be electrostatic, that can be physical, and so forth. And how do you demonstrate sameness with respect to the matrix and how it relates to performance is one of the key areas of complexity we have to deal. Pharmaceutical regulations, pharmaceutical agencies are essentially designed around complexity, but that classification is instinctive. I think when you go back and reflect on it, the organizational means to address complexity, like Center for Drugs versus Center for Biologics, um, often lag behind the products that come through such systems. And you have to be flexible enough to deal with uncertainty and complexity as new products, material analytics come through the system or the organization. The way to think about this is um, what is the difference between an organizational's functioning in a complex versus complicated versus simple product development practice? In complex product development, you need emerging practices. Now, I'm referring to complex in terms of the evidence criteria needed to show sameness. Now, the product we are going to develop as a generic, complex generics, or a biosimilar is perhaps 20 years old or 15 years old. I mean, and so the product itself may no longer be complex, but the pathway to getting it approved is complex now. And that means I'm going to use new analytical characterization, which were not available perhaps to the originator when they were developing their product. Their products were approved primarily on the basis of the two pivotal clinical phase 3 pivotal clinical trials. Therefore, in many aspects, development of a generic or a biosimilar, and to demonstrate sameness using modern analytical tools for physicochemical and biological characterization is a more difficult task than the development of the formulation in the first place that occurred 15, 20 years ago. That's what we have to think about. Now, the difference between complex and complicated is, for complicated, good practices work. Good practices include things like standard operating procedures, project management, quality policy, and so forth. For simple products, you simply need best practices. So what's the difference between complicated versus simple? For complicated processes, you need appropriate education, training, and experience for people to work in the sector that will be able to utilize the SOPs correctly and so forth. For simple, that education, training, and experience is less important. So all pharmaceutical products, once approved, have to have reduced their complexity to a level that they're complicated, so that you have the right education, training, and experience, and the SOPs and the procedures to deliver the product in a consistent, reliable fashion. So the work at R&D has to be is to reduce the complexity by the time the approval is achieved to a level of complicated. So in complex world, given the interaction, given the predictability, lack of predictability and emergent properties, you have emerging practices. You cannot cut and paste and say that the timeline to exhibit batch is this, the timeline to bio batch is this, the way typical generic companies work in their day-to-day routine. So, in that sense, I think it's also important to understand the reasons why FDA defines certain products as complex generics. Primarily, it deals with the difficulty of demonstrating sameness. Many products are locally acting, where the tissue concentration, the site of action is local, and measurement of blood levels is measuring drug in a systemic circulation, which may not reflect the site drug at the site of action. And other reasons for complexity is you often need a clinical endpoint, bioequivalence trial, to be able to integrate all the pieces of evidence, because um, our pharmaceutical equivalence criteria now is really dated. I mean, the laws and regulations written 20, 30 years ago never anticipated the importance of pharmaceutical equivalence and how it will become a dominant force in the equivalence assessment that we need now today. So, sameness determination, or inability to have sameness determination, is one key challenge. The materials can be complex by virtue of, in terms of the analytical characterization. Plus, you have complex patient interactions where and there is a drug device combination, such as dry powder inhaler, where where the key to automatic substitution is the confidence that there is no need for training of patients on how to use the new device. There's no medical intervention needed, is sort of a standard way of thinking about that challenge. So the mechanism of drug device combination, the procedures of using that literally have to be identical. So there is a whole host of maneuvering around the intellectual property and trademark related challenges that exist. Now, to put this in context, I think it will be useful for us to remember. I think quality assurance is a critical quality attribute. As over the years, the number of warning letters, import alerts have increased foreign source material in the US, the level of confidence in the pharmaceutical global pharmaceutical supply chain in the US has diminished to the extent that starting in 2013, 14, 15, the apprehension the patients in America have felt about imported medicine has increased, and there are growing evidence to say that the placebo nocebo effects have become very dominant generally, but also in case of generic. And FDA had to raise a red flag through its guideline, emphasizing that the color, the shape, size of products, generic tablets and capsules matters. And so you can see the patient interaction is also a source of complexity in the whole design process. So just to take a moment here to get our complex and complicated definitions more clear. Lovenox, enoxaparin sodium NDA was approved in 1993. Enoxaparin sodium, Sandoz application was approved in July of 2010. Which of those two products was more complex to develop? If you think about formulation development, API development, focus on that aspect. Which of those two were more difficult to develop? I would pose to you that the Sandoz similar generic Sandoz Momenta product, which was generic to Lovenox, was more difficult to develop because of the most modern orthogonal set of analytics that were utilized to demonstrate sameness. If you don't agree, hold your thought, because I think this is important. The point I'm making here is formulation development of generics, particularly complex generic, is a more difficult, more complex task than the development of the same originator product 10, 15 years ago. And I hope you can appreciate that uniformly and consistently, because we often underestimate that. So my experience was clearly at a high level from senior management perspective to lead the efforts of Omnitrope, Zarzio, Binocrit, and then lay the foundation for the Sandoz monoclonals before I left Sandoz. And I think my experience at Philip Morris International, I think you will hear more of their product as ICOS, which has been launched in Japan, and they have submitted an application to FDA, US FDA, on modified risk tobacco product application. Given for this product, we were working simultaneously as the FDA Center for Tobacco was being established, and we were working before any guideline was developed, before even regulations were outlined. So we had to take a very different approach, and I think this, in my experience, was the most complicated, no, more complex product development program that I had undertaken. And the goal was to bring it to the level of being complicated. And I think time will tell the next few years with the approval, whether FDA thinks that the complexity has been reduced, the evidence is solid to support the product to be launched in US. In Japan, the regulatory framework is different, so the product is very successful in Japan as we speak. As as we think about emergent properties and complex generics, I think I just want to remind ourselves my context remains US FDA. Emergent properties is a hallmark or a clear reminder of a complex system, can have significant impact on development cost, time, and risk of development failure. So as complexity increases, the level of uncertainty can be acceptable to high, depending on whether and draft FDA guidance is available or not. Here, my approach at Sandoz and at Philip Morris was to be at the doorsteps of FDA before they have issued a draft guideline. The reason for this is FDA reviewers are scientists. They come from different backgrounds, and often they will not have the technical expertise necessary to understand all products and to address all level of complexity. So since my firm belief has always been is if you are going to develop a product, you should build yourself to be the expert in that product. Nobody else knows your product better than you do. So if you know your product better than nobody else, who else can then define the sameness as rigorous? The question then becomes, can you defend it? Can you defend it in a way that will convince the regulators to approve the products? So the emergent properties change before issuance of a draft guidance and after issuance of draft guidance. Evolving criteria for evidence needed to establish equivalence is the key concern there. And after issuance of the draft guidance, evolving interpretation of the weight of evidence on effectiveness of efforts to reduce state of complexity to a level of complicated to allow utility of CGMPs are the two emergent properties that you have to address. I have listed certain examples here, case examples. These are the ones I have been involved with as an example. And so I have presented on those before, so you are most welcome to download my slides from my SlideShare account as shown below. And I just wanted to sort of invoke in your mind sucralfate suspension. Now, sucralfate generic drugs as tablets were approved when I was in FDA. It was 2003, around 2003, and there was a significant clinical endpoint study needed. Now we are dealing with sucralfate suspension. As I think about this, how would you define the complexity of sucralfate suspension in terms of generic development and evidence of sameness? Can you think about ways to justify that new clinical study or clinical study with bioequivalence endpoints are no longer necessary? I think that's what the whole theme is all about, and I think you should be able to do that in a pretty predictable way now. So let's think about this bit more. Development initiated before draft guidance. Rich learning opportunities from the information available on enoxaparin, glatiramer, human growth hormone, mometasone nasal spray. I think please look over how FDA has addressed the citizen petitions before approving these products, and as well as what FDA has written in terms of these approvals. Say, for example, the Nature Biotech article on enoxaparin approval, mometasone furoate first approval, the paper that is listed there. And the question I'll ask the audience, I hope you can think about this. Apotex received the first approval for mometasone furoate monohydrate nasal spray generic. I think Amneal got the second approval. All of them had done clinical studies. Now suppose if you are starting the development of mometasone furoate monohydrate nasal spray application now, would you do a clinical study? I hope you'd not, because you have enough learning there to put together your evidence package that the clinical study is no longer necessary. I hope that you see that's the opportunity that we have now. So the challenge that I see around in my practice is many companies are struggling. All of a sudden, as the CMC review moved in with together with Office of Pharmaceutical Quality, and the new drug and the generic CMC review is occurring side by side. The types of queries, the reasons for complete response letters have really dramatically changed. And here is my categorization of that. I think we saw, because of GDUFA related backlogs, a lot of submission issues, administrative refuse to file. I'm not going to talk about those. Those were administrative playing the numbers game that FDA did, I think. But I think the key here is FDA has started asking additional information, many more questions, from polymorphism, control of polymorphism, control of particle size, shape, and charge, to the old ways of revising specification after the product has been developed. That's unfortunate. That continues. But equally, I think important is FDA is no longer really open to the 10x scale up criteria unless you have proved through your development program that your manufacturing process is well understood and controlled and the scale up is justifiable, especially for complex generic products. So all of those things point to a very clear message, and that being the quality by design paradigm has existed for a very long time, and the methodology for explaining how quality was built into the product by design was outlined in 2005 through ICHQ 8, 9, and 10, 11, and so forth. Adoption and practice of QBD has seriously lagged behind. I see literally everybody talking QBD, 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 and I repeat myself here. And for me, that's kabaddi, kabaddi, kabaddi. And we really need to take a deeper dive to understand what are the scientific questions, what are the assumptions that are okay to keep, and what are the assumptions that need extra validation, extra verification. But what are the key scientific questions, and how do you communicate the steps in formulation development that you have taken as a logical scientific progression that keeps reducing residual uncertainty? That is an element of scientific writing that many companies are lacking. I think most of the scientific writing that occurs on regulatory submission has become, in my way of thinking, a best practice of cut and paste. The cut and paste best practice doesn't work in a complicated good practice, and hopefully the complexity has been reduced. So keep that in mind. Effective communication is a key. Now, at the same time, as an ex-FDA, I have been concern for all my career to that extent is, I think, the timing of generic drugs, and the pressure on generic drugs is high on the companies as well as on FDA. What do I mean by that? What I mean by that is Hatch-Waxman rightly incentivizes first generic, and there are provisions to file first, and there is a economic incentive for doing that for 180-day exclusivity. But the practice around that has become file first and hope to figure it out later, and that, I believe, is a challenge. So we need to find a way to file first and figure it out at the time of filing. That means there has to be a bit more early investment in the development process. And I want to talk about that in a minute. I think OGD's exuberance in telling everyone that, oh, we are achieving quality by design 80% or so through question-based review was a bit immature. And I think today you're seeing that the quality by design elements, really important aspect of that is in measurement and control and a state of control of the manufacturing process. But at the same time, I think an integrated assessment of the totality of evidence and the weight of evidence. FDA itself struggles in that way of how do you properly weigh evidence, in vitro PK, clinical endpoint bio study, and I think that will remain uncertain until for a foreseeable future. So when we see regulatory queries, are they expected or unexpected? Given the experience, given the range of products I have seen, and given the leadership role I have had in shaping the 21st Century Initiative at FDA, for me, about 90% of those queries are completely expected. They're going in the direction of the 21st century quality by design. OGD may not have asked, or CMC reviewers may not have asked that before, but they're expected. 10% of those queries may not make sense. They are reflecting some of the minute aspects that are pertaining to a reviewer's own expertise, but may not have big relevance in the overall process. They're not risk-based. So I think you have to find a way to expect the expected and the unexpected. And I think so. There is an important research component. Remember, I think you guys, I'm saying you guys, all of formulation development is part of research and development. And there is an important emphasis on research that we should not forget. We jump into development as if it's a cookie cutter, cut and paste. That's not what this development is. There is a significant research component that we need to talk about. And that research component really is informing on your target product profile and your quality target product profile. And that is not the way a short table. It is an in-depth understanding of what are the critical attributes, what are the failure modes, and what is a portfolio of analytical characterization tools that allow you to reduce uncertainty to an extent that you can judge whether a clinical endpoint study is needed or not. That's what has to go into research, along with your IP and other considerations that are important. So I think an emphasis on research is necessary. In my experience, for a typical modified release dosage forms, I have seen companies successful in getting an approval in 24 months, very rarely, but I have seen that. But on the longer side, it's taking many companies 42 months. So where do you stand in a typical modified release dosage form type scenario, and what will your expectation be for a more complex generic? I think those are important considerations. So let me sort of summarize my thoughts. Uh, in the 21st century, pharmaceutical quality for the 21st century heavily emphasized science-based development, science of manufacturing, so that we can make risk-based decisions. We don't make risk-based decisions today because our information is, the scientific rigor of our information does not provide the level of objectivity we need to make risk-based decisions. So science is going to be important, and science means there's a theory. You test the hypothesis using validated methods, and without theory and without having a hypothesis, it's very difficult to call yourself scientific. Therefore, you have to think about your formulation development, your analytical characterization in the context of what is your hypothesis, what are your questions, and why do you think your methods are validated. In the typical generic scenario, method validation occurs way late in the development process. In complex generics, biosimilars, method validation, especially the ones which becomes part of the pivotal in vitro evidence, has to be validated first. You don't spend your time doing a lot of characterization using methods which are not validated. That's fundamental. And I think that mindset has to change. As we approach the year 2020, I believe science of formulation and manufacturing will be the dominant part of good practices. This will be profound. Complexity will continue to increase, requiring rigorous science practice. We have to anticipate how criteria for evidence needed to establish equivalence will evolve. If you're willing to take the chance and go to FDA for a product where FDA has not even put a draft guidance yet, anticipate what would be the blocking citizen petition and how would you overcome that citizen petition and build that into your development program. And that then becomes a favorite application that FDA will have to use to address the citizen petition that will come, and you get first to market that way. Interpretation of weight of evidence requires each piece of evidence to be scientifically rigorous. When you design a clinical study, when you design a PK-based bioequivalence trial, the statistical aspect of those trials are well founded. Nobody can tell you that they're not scientific anymore. You have to bring the same thinking, statistical rigor, test of hypothesis to your in vitro characterization so that the weight of that evidence is clearly evident. Without that, there is no chance that you can have a request for waiver of a bio study or a clinical study. So put your emphasis on weight of evidence utilizing scientific test of hypothesis of your in vitro characterization data. So you need to conduct research to understand your and quantify your quality target product profile. This is, in my way of calling it, is a new prior knowledge. You are going to use the reference product, characterize it with the most modern analytical characterization tools. You will characterize attributes that perhaps the originator never characterized. Say, for example, when we talk about dry powder inhalers, you're characterizing aspects such as shape, size, static charges, binding of materials in a way that you have to build more knowledge on that perhaps the originator had. And you have to then put this in the context of a scientific test of hypothesis. This is very important. So new prior knowledge, you have to understand your failure modes, and you have to design your quality target product profile, which is far more detailed than the few bullet points are, as in the FDA example on their website. That's not really what is needed. You need to understand the critical quality attributes of your RLD, and the acceptable variability by lot to lot variability, and you have to design your formulation and your manufacturing process so that your product is consistently within that range of lot to lot variability of RLD. The sooner you can invest in RLD characterization, the wiser you will be in, and the more benefits you can derive. I think on my last slide, I will summarize my thoughts here. I think $1 spent on R&D in most Indian companies have over the years given $9 in revenue. That's traditional. I think $10 spent on R&D for complex generics can give you $150 or more of revenue. That's what we have as an opportunity, but that's also you have as a risk. You have the risk of losing your $10 investment more easily if you don't think about and appreciate the complexity and the science of complexity and how do you leverage that complexity. And the way to do that is initial research, new prior knowledge to set up your hypothesis of what is critical, what is necessary for sameness, and emphasize in vitro to the extent and show through in vitro analytical characterization what is the residual uncertainty, so that, and if you have to do any clinical or in human studies, you're focusing those studies on that residual uncertainty. And then you have to put together your scientific evidence package in an integrated manner, not a cut and paste way. So quality, target, product profile, key questions and assumptions have to be tracked, and your residual uncertainty has to be understood very clearly to say, do we need any more testing or not? I hope this information will be of use to you. You will find this useful, and I really thank you for your attention. Thank you.